In this segment, we will discuss how to locate the promoter in a gene. Now, in principle, class 2 promoter, they should be easier to locate than the enhancer. That is the principle, that the class 2 promoter, they should be easier to locate the enhancer. Why? The reason is because class 2 promoters, they lie at very near the transcription start sites of genes which are known. So they are lying at a very start of the transcription sites. Now, Bingrang and colleagues, they performed a genome-wide search for human promoter. They wanted to know that how many human promoters are there and where they are located. They found that many genes have alternative promoter that are located hundreds of base pairs away from the primary one. So the one thing which they found important is that many promoters, they have alternative promoters which were situated at hundreds of base pair away from the primary ones. So they searched for the promoter in human fibroblasts using a chip strategy. That was the strategy which we have already studied. Now, these scientists, they performed chip using a monoclonal antibody against a TAF1 subunit of TFIID. Now, why they used that antibody? Because the pre-initiation complexes which were forming at the promoter, that should contain this key general transcription factor. That was their hypothesis, and that was the reason they used that antibody. Now, they amplified the DNA precipitated by chip and used it to probe DNA microarray 14.5 million to 50 MERS size, which was there. Now, TFIID binding sites, there were 12,150 binding sites were found, of which 10,553, that is the 87.87%, that was mapped within 2.5 kilobase of a known transcription start site. So the transcription start sites which were of 2.5 kilobase, that was found within that region. Now, why they use 2.5 kilobase start area is that they had to use the fairly large window of 2.5 kilobase to allow for uncertainty in the mapping of transcript of 59 ends. The 59 end transcript they had to map. Now, TFIID binding sites were mapped, and these binding sites, they were mapped to the same transcript, that is the 59 ends, by eliminating these redundancies. There were those redundancies which were present, they eliminated those redundancies, and these sites that was settled on 9,328 binding appear to a unique transcript. That is, of these 9,328 binding, they appear to the unique transcripts in this. And there were 4 tests for promoters like characters. That's how we can find out that these promoter characters, they are there. The first test was that the chip-chip analysis was performed with an anti-RNA polymerase II antibody. It was found that 97% of the TF-IID binding sites also bound polymerase II. So that was the test which was there. When the second was the 28 sites were selected and the standard chip analysis was performed with an anti-RNA polymerase antibody again of the 28 sites. And thirdly, CPG islands and the INR, DPE and the Tata boxes core promoter elements in the 9328 TF-IID binding sites. They were searched in that by what? CPG island, DPE and the Tata boxes. So what they found? They found that the enrichment for the first 3 but not for the Tata boxes. There was no enrichment of these promoter in the Tata boxes. Now, the fourth chip analysis that was used to look for histone modifications. That is the acylated histone H3 and dimethylated lysine 4 on histone 3. So these were the 4 tests which were performed to find out the promoter location in the human fibroblasts.